So we're going to be finishing up the third part for Chapter 11. This is Part C. And be sure to watch the first two videos if you haven't already, Part A and Part B for the nervous system and nervous tissue. You can find the link for those in the description below. As always, if you like this video, please be sure to give the thumbs up. And also be sure to subscribe and let your friends, your classmates know about this videos. The nervous system's ability to function properly is dependent upon the flow of information from neuron to neuron. And it's at the synapse, which is a junction that mediates the information transfer from one neuron to another neuron or from a neuron to an effector cell. Presynaptic neurons are the neurons that are conducting impulses towards a synapse, so they're sending information. When you look at this term right here, presynaptic, this prefix pre means before. So again, these are the neurons that come before the synapse, and the electrical impulse is traveling away from the cell body, so it's sending information out of this neuron. And when you look at the term postsynaptic, post means after the synapse. So postsynaptic neurons are the neurons that are transmitting electrical signals away from the synapse. These are the neurons that are receiving information. So the electrical signal is coming towards the cell body. And outside of the central nervous system in the PNS, the postsynaptic cell could be that of another neuron, a muscle cell, or a gland cell. And most synapses, they function as both presynaptic and postsynaptic neurons. In this image over here, you can see the axon of a presynaptic neuron here, and there's another one over here. And this right over here is the cell body of a postsynaptic neuron, the soma. When you find the synapse between an axon terminal of one neuron and the dendrite of another, it's called the axodendritic synapse. When you find that between an axon terminal of one neuron and the cell body of another, or the soma of another neuron, that's called an axosomatic synapse. The axodendritic and the axosomatic are the two most common types of synapse we have. The ones that are far less common are the axoaxonal, where we have the axons of the two neurons that are forming the synapse. The dendrodendritic, this is where we have the dendrites of the two neurons that are forming the synapse. And then we have the somatodendritic. This is where we have the dendrite of one neuron and the cell body of another, or the soma of the other. The two main types of synapses we have are the chemical synapse and the electrical synapse. In this image over here, we can see an axodendritic, axosomatic, and the axoaxonal synapse. So over here, here's the axon of one neuron, and we have the cell body of another. So it's going to be the axosomatic. And over here, we have the axon of one neuron and the dendrites of another. So this is the axodendritic. And then here's, again, another axon of one neuron, and we have the axon of another neuron. So we have the axoaxonal synapse. Between the two axons. Chemical synapses are the most common type of synapse, and they're specialized for the release and reception of chemical neurotransmitters. And they're typically made up of two parts, the axon terminal of a presynaptic neuron and the receptor region on the postsynaptic neuron. On the axon terminal of the presynaptic neuron, it's going to contain the synaptic vesicles that are filled with neurotransmitters. And on the receptor region on the postsynaptic neuron's membrane, it's going to be receiving the neurotransmitters. These are usually found on the dendrites or the cell bodies of the postsynaptic neuron. The pre- and postsynaptic membranes are always separated by a fluid-filled space called the synaptic cleft. So if you remember the chain of events that takes place at a neuromuscular junction that we learned in the chapter on muscles a few chapters ago, an electrical signal gets changed to a chemical signal, which crosses the synapse, and then it changes back into another electrical signal. The current from the presynaptic membrane dissipates in the synaptic cleft. This prevents a nerve impulse from being directly transmitted from one neuron to another. The transmission of signals across the synapse is a chemical event that's dependent upon the release, diffusion, and receptor binding of neurotransmitter molecules. This ensures unidirectional communication between the neurons. Be sure you watch this animation on neurotransmitters. Go to the Pearson website, go to YouTube, or you can refer to the DVD that may have come with your book. Now let's take a look at the 6 steps that are involved in the information transfer across chemical synapses. And you guys may remember most of this from when we learned about the neuromuscular junction. So in the first step, the first thing that happens is that an action potential arrives at the axon terminal of the presynaptic neuron. And then what this does is, number 2, is that it's going to open up these voltage-gated calcium channels. So what it ends up happening now is that you get an influx of calcium to enter the axon terminal. So calcium is going to be flowing down its electrochemical gradient from the extracellular fluid into the inside of the axon terminal. So when you look over here, Here's your axon terminal. This is the presynaptic neuron. And as this action potential is coming down, it causes these voltage-gated calcium channels to open up as well. Now, we have sodium-gated channels here too, but among them, the area of interest right now are these calcium-gated channels. So when these open up, all the calcium that's outside is going to end up coming into this axon terminal through these voltage-gated calcium channels. So remember, what calcium is doing is going down its concentration gradient. In the third step, the calcium entry is causing the synaptic vesicles to release the neurotransmitters. So what's happening is that the surge of calcium that's coming into the axon terminal, it acts as an intracellular messenger. There's a calcium-sensing protein called synaptotagmin that binds to the calcium, and it interacts with the SNARE proteins that control membrane fusion. Fusion will result in exocytosis of the neurotransmitters into the synaptic cleft. Now, the more intense the stimulus is, the greater the number of synaptic vesicles are going to fuse and spill their contents, and this is going to cause a greater effect on the postsynaptic cell. So in this image here, we're going to be going over the first 3 steps that we just talked about. So what we have over here, what we're looking at is this presynaptic neuron, and then this is the postsynaptic neuron. So the action potential is going to be traveling down this axon. Then it comes to this axon terminal. Now over here in the axon terminal, what do we have here? We have these synaptic vesicles that are filled with the neurotransmitters. Also over here, we have these voltage-gated calcium channels. There's also sodium channels that are not shown, but let's just stick with what we have at this point. So these voltage-gated calcium channels will open as the action potential arrives over here. When these voltage-gated channels open, we have a lot of calcium on the outside, and remember, calcium is going to want to travel down its concentration gradient. So it's going to go from this area of high concentration and enter this axon terminal where the concentration is low. When the calcium enters this axon terminal, it's going to go and bind to those synaptotagmin proteins that we spoke about earlier. When it binds to the synaptotagmin protein, those synaptotagmin proteins, it's going to want to come down over here because it's attracted to these phospholipids in this presynaptic membrane. As it's coming down, it's going to cause the SNARE proteins to bind to these synaptic vesicles. Once the synaptic vesicle binds to the presynaptic membrane, we have exocytosis of the neurotransmitters into the synaptic cleft. In the fourth step, neurotransmitters diffuse across the synaptic cleft and they bind to specific receptors on the postsynaptic membrane. These receptors are called ligand-gated ion channels, or you can also call them chemically gated ion channels. In the fifth step, binding of neurotransmitter opens ion channels resulting in graded potentials. So when the neurotransmitters bind to the receptor proteins, the receptors change their shape. Now this change in shape causes ion channels to open and it creates a graded potential. Depending on the receptor protein to which the neurotransmitter binds and the type of channel the receptor controls, the postsynaptic neuron could be either excited or inhibited. We also see postsynaptic membranes that often contain receptor proteins and ion channels that are packaged together, and they create these chemically gated ion channels. So in this slide here, we're going to be going over the fourth and fifth steps. So here we have these neurotransmitters. They're moving across the synaptic cleft, and now they come and they bind to these ligand-gated ion channels. So once they bind, it causes a change in the shape of these proteins, and now we end up having an influx of ions into this postsynaptic membrane. And this is what's going to cause the graded potential in the postsynaptic membrane. So you can see that over here as being the fifth step. In the sixth step, the neurotransmitter's effects are terminated. As long as the neurotransmitter is bound to the postsynaptic receptors, the graded potentials will continue. In addition to that, it's going to affect the permeability and block the reception of additional signals from the presynaptic neurons. And it's for this reason the process needs to be regulated. The neurotransmitter's effects usually last a few milliseconds before being terminated in one of 3 ways. Depending on the particular type of neurotransmitter, it can occur through the reuptake by astrocytes or the presynaptic terminal where the neurotransmitter is stored or destroyed by enzymes, as we see with norepinephrine. It can also be terminated through degradation by enzymes that are associated with the postsynaptic membrane or present in the synapse, as is the case with acetylcholine. Finally, termination can also occur through diffusion away from the synaptic cleft. And in this slide here, we want to look at step number 6, where the neurotransmitter's effects are being terminated. And as we've just learned, this can happen through one of 3 ways: by enzymatic degradation, by diffusion away from the synapse, or through the reuptake by the astrocytes. As we learned in the previous lecture that action potentials can travel at speeds of up to 150 meters per second down an axon. However, neurotransmissions across a chemical synapse is comparatively slow, and this is because of the time that's required for the neurotransmitters to be released, for them to diffuse across the synaptic cleft, and for them to bind to the receptors. This usually lasts anywhere from 0.3 to 5 milliseconds, and it's called the synaptic delay, and this is what's the rate-limiting step of neural transmission. So now you can better understand how fast the action potential occurs down the axon, but when we get to the synapse, it slows the transmission to the postsynaptic neuron significantly. At the same time, it's not very noticeable because it's still happening very fast. When you look at the action potential that's traveling down the axon at 150 meters a second, 0.3 to 5 milliseconds is a very small period of time. So the entire process is still occurring very fast. Now we're going to be looking at electrical synapses, and these are much less common than chemical synapses. These neurons are said to be electrically coupled because of the protein channels called connexons that are connecting the cytoplasms of the adjacent neurons. These channels, they allow the ions and small molecules to flow directly from one neuron to the next. Transmission across these synapses occurs very quickly, and depending on the nature of the synapse, communications may be unidirectional or bidirectional. We find electrical synapses in the region of the brain that's responsible for eye movement, or the hippocampus, which is involved in emotions and memory. We find them most abundant in embryonic nervous tissue, where they allow for the exchange of guiding cues during early neuronal development, so that the neurons are able to connect properly with one another. Unlike the voltage-gated ion channels that are responsible for action potentials, and chemically gated channels, the neurotransmitter receptors cause graded potentials that vary in strength based on the amount of neurotransmitter that's released and the time that neurotransmitter stays in the cleft. And depending on the effect of chemical synapse, there are two types of postsynaptic potentials. There's excitatory postsynaptic potentials and inhibitory postsynaptic potentials. When the neurotransmitters bind to the chemically gated ion channels, the gates open. This allows for the simultaneous flow of sodium into the cell and potassium out of the cell. Now remember, the electrochemical gradient for sodium is much steeper than it is for potassium. So the sodium influx is going to be much greater than the potassium outflux. Now this results in a local net graded potential depolarization that's called the excitatory postsynaptic potential, or EPSP. If enough neurotransmitters bind and depolarization of the postsynaptic membrane reaches its threshold, then an action potential will be generated. While the currents that are created by individual ESPSs decline with distance, they can and they often do spread all the way down to the hillock. And if the currents reaching the hillock are strong enough to depolarize the axon to threshold, axonal voltage-gated channels open and an action potential is generated. And in this slide here, we're just going over what we learned in the past couple of slides. So remember, an EPSP is a local depolarization of the postsynaptic membrane. And it's trying to bring the neuron closer to the threshold, to the AP threshold. So if we meet the threshold, then the EPSP is going to travel down, the action potential is going to travel down to the axon hillock. And then if it's a strong enough current that reaches this axon hillock, and it's able to depolarize the axon threshold, then the axonal voltage gate channels will open and an action potential is going to be generated that's going to travel down the axon. The majority of inhibitory neurotransmitters induce hyperpolarization of the postsynaptic membrane by making the membrane more permeable to potassium ions or chloride ions. If the potassium channels are open, potassium is going to move out of the cell. If the chloride channels are open, then chloride is going to move inside the cell. Either way, the charge on the inner face of the membrane becomes more negative. And as the membrane potential increases and is driven further away from the axon's threshold, the postsynaptic neuron becomes less likely to fire an action potential, and larger depolarizing currents are required to induce an action potential. These changes in potentials are called inhibitory postsynaptic potentials, or IPSPs. So this image over here in this slide goes over what we just learned in the previous slide. So again, IPSP, So this image over here goes over what we just learned in the previous slide. So remember, IPSP stands for inhibitory postsynaptic potential. And essentially what it's showing is that when inhibitory neurotransmitters attach themselves onto the postsynaptic membranes, it's going to cause these chemically gated ion channels that are permeable to either potassium or chlorides to open up. The efflux of potassium out of the cell and the influx of calcium into the cell is going to hyperpolarize the resting membrane potential. So you can see over here, it starts out from minus 70, and when these gates open, it goes on even further more. So now maybe it's at this minus 90 point. An EPSP by itself is not able to induce an action potential in the postsynaptic neuron. But if thousands of excitatory axon terminals are firing on the same postsynaptic membrane, or if a small number of terminals are delivering impulses rapidly, the probability of reaching threshold depolarization increases greatly. So what this means is that the ESPSs have an ability to add together or summate to influence the activity of a postsynaptic neuron. Most neurons that receive both excitatory and inhibitory inputs from thousands of other neurons, and only if the EPSPs they predominate and bring to threshold will we end up getting an action potential that's generated. And we have two types of summations. We have temporal and spatial summation, and we're going to be taking a look at both these in the next few slides. Temporal summation occurs when one or more presynaptic neuron transmits impulse in a rapid-fire order, and bursts of neurotransmitters are released in quick succession. So the first impulse produces a small EPSP, and before it can dissipate, another EPSP is triggered, adding on top of the first impulse. These summate or add up, producing a much greater depolarization of the postsynaptic membrane than we would get from a single EPSP. In a spatial summation, the postsynaptic neuron is stimulated at the same time by a large number of terminals from the same, or more commonly, different neurons. And we get a huge number of its receptors that bind neurotransmitters and simultaneously initiate EPSPs. This summates and dramatically enhances the depolarization. And the other thing to mention, which is not listed over here, is that IPSPs also summate both temporally and spatially. So in their case, the postsynaptic neuron is going to be inhibited to a greater degree. So when you look at this slide over here, this dashed line represents the axon threshold, and this solid line over here is the resting potential. So this shows the membrane's potential in millivolts, and this shows time. And what we have over here is a single excitatory synapse, okay, which is represented by E1. So you have E1 here, E1 here. So this is the same synapse. Now the stimuli occurs far enough in time that it doesn't allow for a summation to reach the threshold of the axon, which is over here. And in this slide here, this is an example of temporal summation. So we still have a single excitatory synapse. However, the stimuli, they occur close enough in time that they build upon and they exceed this threshold. So in this slide, we're going to be looking at this spatial summation. So we have two excitatory synapses here. We have one over here and we have another over here. So when both these guys fire, it's through their summation that the EPSP is able to add together and exceed this threshold onto this neuron over here. So in this image over here, we have both an IPSP and an EPSP. So when you look at this graph here, when the IPSP sets off, we end up getting hyperpolarization. So remember, this is the resting membrane potential of -70. And so when we get the stimulus over here, we go underneath or we go below, because remember, this is going to be an inhibitory synapse. Now, when we go over here to the excitatory synapse, okay, the EPSP, this is going to end up bringing it back up. However, when you look at the summation that you get over here versus what you get over here, they end up canceling one another out. So now you end up going back to this resting membrane potential of -70. Repeated or continuous use of a synapse, even for a short period of time, enhances the presynaptic neuron's ability to excite the postsynaptic neuron. This produces larger than expected postsynaptic potentials. And this is what's called synaptic potentiation. So these presynaptic terminals at these synapses contain a relatively high calcium ion concentration, and this condition triggers the release of more neurotransmitters, which in turn produces larger EPSPs. So again, notice this is an error. This should say larger, not more. Synaptic potentiation brings about a calcium influx into the postsynaptic neurons as well. The brief high-frequency stimulation partially depolarizes the postsynaptic membrane. Now, this partial depolarization is key as it causes certain chemically gated channels called NMDA receptors to allow calcium entry. As calcium ions flood into the cell, they activate certain kinase enzymes which promote changes that result in more effective responses to subsequent stimuli. Synaptic potentiation can be viewed as a learning process that increases the efficiency of neurotransmission along a particular pathway. The hippocampus of the brain plays a special role in memory and learning. It exhibits an important type of synaptic plasticity called long-term potentiation. Presynaptic inhibition occurs when the release of excitatory neurotransmitters by one neuron is inhibited by the activity of another neuron via axoaxonic synapses. While several mechanisms are involved, the end result is that less neurotransmitters are released and bound and smaller ESPSs are formed. So when we look at this slide here, it's a comparison of graded potentials and action potentials. So what we're looking at over here is multipolar neuron, and they're telling us that for graded potentials, they occur in the dendrites and the cell bodies, typically. And for action potentials, they occur at the axon hillocks, where they're going to then travel down the axon. And for the ones that are originated in the dendrite and cell body, they're going to travel to the axon hillock. So, which leads us to the next part, which is distance traveled. So, the graded potentials, they travel a relatively short distance, anywhere from 0.1 to 1 millimeter. So again, it's going from a dendrite or somewhere in the cell body to the axon hillock. And for the action potential, it will travel from the axon helix all the way down the entire length of an axon, and this could be a few millimeters to over a meter in length. Continuing with our comparison of graded potentials and action potentials, the amplitude size is going to vary in graded potentials, and as the distance increases, the amplitude will decay. In action potentials, however, it's always going to be the same size because of the all or none phenomenon. It does not decay with distance. As for the stimulus for opening ion channels, for graded potentials, it's going to be chemical, so the neurotransmitters that we just learned about. In addition to that, it can also be sensory stimulus such as light, pressure, or temperature. As far as summation goes, it doesn't occur in action potentials. Instead, we have the all or none phenomenon. In graded potentials, we have temporal and spatial summation. In temporal summation, it's from the increased frequency of the stimuli, whereas in the case of spatial summation, the stimuli comes from multiple sources. For the graded potentials, we have two types of postsynaptic potentials. We have the EPSP, or the excitatory, and the IPSP, which are the inhibitory. For the EPSP, these are short-distance signaling. Its depolarization spreads to the axon helix, which moves the membrane potential towards the threshold for generating an action potential. IPSPs are the opposite of EPSPs. Instead of depolarization, we have hyperpolarization that spreads to the axon helix, and it moves the membrane potential away from the threshold for generating an action potential. And for action potentials, we have long-distance signaling, which constitutes a nerve impulse. As far as the initial effects of stimulus in the graded potentials, in the EPSP, we have these chemically gated channels that allow the simultaneous flow of sodium into the cell and the flow of potassium out of the cell. For the IPSPs, we have channels that are separate for potassium and for chloride ions. So we have potassium ions that are exiting the cell, and we have chloride ions that are entering the cell. In action potentials, we have these voltage-gated channels. So first we get this sodium voltage-gated channel that opens up, and it allows for the influx of sodium ions. And next, we have the opening of the potassium-gated voltage ion channels, which allows for the efflux of potassium out of the cell. When we look at the peak membrane potential in the graded potentials, when it's depolarizes, it moves towards 0 millivolts, and when it's hyperpolarizes, it moves towards minus 90 millivolts. When you look at the action potential, it goes from, this should say minus, not positive. It goes from negative 70 to positive 30, and in some cases, positive 50 millivolts. Along with electrical signals, neurotransmitters are the language of the nervous system. This is the means by which each neuron communicates with others to process and send messages to the rest of the body. More than 50 neurotransmitters have been identified so far, and while most neurons produce and release only one kind of neurotransmitter, most of them, they make two or more, and they may release any one or all of them. It appears that in most cases, different neurotransmitters are released at different stimulation frequencies, which is a restriction that avoids producing nonsense messages. There has been documentation of the co-release of two neurotransmitters from the same vesicle. The coexistence of more than one neurotransmitter in a single neuron makes it possible for that cell to exert several influences rather than one discrete effect. We classify neurotransmitters by their chemical structure and their function. Acetylcholine, or some people also call it acetylcholine, you can say whichever one that you want, doesn't make a difference. People will understand it. This is the first neurotransmitter that was identified, and it's the best understood, primarily because it's very easily found at the neuromuscular junction compared to the ones that you find in your CNS, which are deeply buried and they're much harder to study. So it's released by all neurons that are stimulating skeletal muscles, and also by some neurons of the autonomic nervous system. We also find acetylcholine in some of the neurons that are found in the central nervous system. It's made from acetic acid and choline by the enzyme choline acetyltransferase. And after it's synthesized, it gets sent to the vesicles where it's stored for release. And when it's released, these neurotransmitters will then go bind to the postsynaptic receptors. And at that point, after a few milliseconds, they end up getting degraded back to acetic acid and choline by the enzyme acetylcholine esterase. The liberated choline then gets picked up and sent back to the presynaptic terminal, where it gets recycled to produce more acetylcholine. Biogenic amines include catecholamines and indolamines. For the catecholamines, we have dopamine, norepinephrine, and epinephrine, and these are all made from the amino acid tyrosine. The indolamines include serotonin and histamine. Serotonin is made from the amino acid tryptophan, and histamine is made from the amino acid histidine. They're also released by some of the motor neurons of the autonomic nervous system, especially norepinephrine. Imbalance of these neurotransmitters are associated with mental illness. So for example, overactive dopamine signaling occurs in schizophrenia. We also see psychotropic drugs like LSD and mescaline that can bind to biogenic amine receptors and induce hallucinations. We also see low levels of serotonin being associated with depression. So again, people that suffer from depression, a lot of the times the doctors, they prescribe these drugs called selective serotonin reuptake inhibitors, SSRIs. So this is your Prozac, your Paxil, your Zoloft. These medications, what they do is they inhibit the reuptake of serotonin. So more serotonin is floating around. And if you look at what serotonin is made from, it's made from the amino acid tryptophan. So nowadays, especially, a lot of people, they want to take the natural route. So what they sell at, if you go to Whole Foods, or again, if you go to a naturopath, they sell these something called 5-hydroxytryptophan, or more commonly known as 5-HTP. And what 5-HTP is a precursor to tryptophan. So the thinking is that if you take 5-HTP, then you boost your levels of tryptophan, and then your body's going to convert the tryptophan into serotonin. It works well for some people. It doesn't work well for the others. Best thing to do is go talk to your doctor and let your doctor decide which is the correct course of treatment for you. Of course, the doctor will probably tell you to go with the SSRIs because this is what they're licensed to prescribe. And the FDA doesn't regulate these supplements such as 5-HTP. So there's not really that much research that's done, or it's not approved medically by Western medicine to prescribe 5-HTP. Again, it's an alternative treatment that some naturopaths prescribe, and some people, they go out and they avoid seeing the doctor and they try to treat their symptoms by themselves. I don't recommend that. Again, go and speak with your healthcare professional and let them decide what's best. I'm not saying that it doesn't work. It has worked very well in a lot of individuals. What I'm saying is that you need to be under the supervision of a physician, of a health practitioner, to best decide how to treat and monitor this condition. As far as amino acids go, because they make up all the proteins in our body, it's much more difficult to prove which ones are actually neurotransmitters. However, the ones that are proven to be the neurotransmitters include glutamate, aspartate, glycine, and GABA, which stands for gamma aminobutyric acid, or gamma beta aminobutyric acid. The neuropeptides are essentially a string of amino acids that include a broad spectrum of molecules with diverse effects. For example, the neuropeptide called substance P is a mediator of pain signals. On the other hand, endorphins such as beta endorphin, dynorphin, and enkephalins, these act as natural opiates. So what they're doing is they're reducing the perception of pain. So when you look at a woman that's in labor, the pain that's associated with labor, which is excruciating, it's helped somewhat by the release of these enkephalins. When you look at athletes, for example, a runner who's in a marathon, they've been running, for example, let's just say it's a 20-mile marathon. So, they end up doing really good, the first 5 miles, the next, 5 miles, okay. The 5 miles after that, they start to get tired down at that point. And, if you look at their face or their, some of their body language, you might sense that they're uncomfortable, that, that they're in pain, but they continue to go. And then at some point, you might even see probably the last couple of miles or last mile or so, you'll know, especially in trained athletes, you'll notice that they end up getting this rush. And this rush is what we call this runner's high. And what the body is doing is releasing this surge of these neurotransmitters, specifically this beta endorphin. And it gives them a little bit more of energy, and it causes them to go even a little bit more faster. Their sense of pain, it kind of just all goes away. So what ends up happening is they end up getting this sense of euphoria, this false sense of euphoria. Because the reason I call it a false sense is, your body is hurting at that point. They're starting to get dehydrated, especially if they're not drinking enough liquids, if they're running throughout the marathon. So again, the body is under a lot of stress, and them feeling great, it's not real. It's this false sense, and your body is just trying to get you out of that situation. So moving on, other neuropeptides such as somatostatin and cholecystokinin, these are also produced by non-neural body tissues, and they're widespread in the GI tract. These peptides are commonly referred to as the gut-brain peptides. Moving on to the purines, we have adenosine triphosphate, or ATP, and we've learned in previous chapters that ATP is a universal form of energy. We now know that it's also recognized as a major neurotransmitter in both your central nervous system and the peripheral nervous system. Depending on the ATP receptor type it binds to, ATP can mediate fast excitatory responses, or it can trigger slow second messenger responses. And upon binding to receptors on astrocytes, it mediates the influx of calcium. Aside from the neurotransmitter actions of ATP, adenosine, which is part of ATP, also acts outside of the cell on adenosine receptors. Adenosine is a potent inhibitor in the brain, and caffeine's stimulatory effects results from its block of these adenosine receptors. Moving on to gases and lipids, not too long ago, gases like nitric oxide, carbon monoxide, and hydrogen sulfide were never thought to be beneficial or part of the body. These were always thought to be toxic, and they are highly toxic gases. However, we now know that our body actually uses these gases as neurotransmitters. They actually use them as messengers. These gases are lipid soluble, so they're easily able to pass through the plasma membrane. They're also synthesized on demand, so they're not stored and they're not produced and then stored in vesicles. Instead, when your body needs it, they're produced on demand and then they get shipped out of the cell. And then they'll pass through the plasma membrane of the nearby cell and bind to the specific intracellular receptors of that second cell. Nitric oxide is involved in the learning and formation of new memories by increasing the strength of certain synapses. On the other hand, excessive release of nitrous oxide excessive release of this gas results in brain damage, as we see in stroke patients. It also causes the smooth muscles of your intestines to relax. And hydrogen sulfide gases will directly alter ion channel function. Endocannabinoids, they act at the same receptors as THC, which is an active ingredient in marijuana. This is a recently discovered class of neurotransmitter, and we now know that the receptors are the most common G-protein coupled receptors in the brain. And like nitric oxide, they're also lipid soluble and they're synthesized on demand. They're also believed to be involved in the learning and memory process. We're still learning a lot about endocannabinoids, but we think they may be involved in neural development, controlling appetite, and suppressing nausea. Although neurotransmitters have a very great diversity of functions, we're going to be limiting our scope and looking at two things. We're going to be looking at the effects and actions of neurotransmitters. Now, the effects of the neurotransmitters is going to be either excitatory or inhibitory. In other words, either depolarizing or hyperpolarizing. Now, some neurotransmitters, they can do both things depending on the receptors to which they're attached to. For example, GABA and glycine are usually inhibitory, whereas glutamate is usually excitatory. Acetylcholine and norepinephrine, on the other hand, they bind to at least two different types of receptors that have the opposite effects. So when acetylcholine is bound to the neuromuscular junction, it's going to have an excitatory effect, as we already know. But when it binds to cardiac muscle, it has an inhibitory effect. For neurotransmitter actions, they're either direct or indirect. Neurotransmitters that bind to and open ion channels are said to act directly. They cause a rapid response in the postsynaptic cell by altering the membrane potential. So acetylcholine and amino acids are direct-acting neurotransmitters. Neurotransmitters that act indirectly tend to promote broader, longer-lasting effects by acting through intracellular second messenger molecules. Typically, this is via G-protein pathways. So these indirect neurotransmitters include biogenic amines, neuropeptides, and dissolved gases. Neuromodulator is a term that's used to describe a chemical messenger that's released by a neuron that does not directly cause ESPSs or ISPSs, but instead it affects the strength of a synaptic transmission. It may act presynaptically to influence the synthesis, release, degradation, or reuptake of neurotransmitters, or it could act postsynaptically by altering the sensitivity of the postsynaptic membrane to the neurotransmitter. Neuromodulator receptors aren't necessarily found at a synapse. Instead, a neuromodulator may be released from one cell to act at many cells in its vicinity in a manner that's typical of paracrines. Paracrines are chemical messengers that act locally and are quickly destroyed. Channel link receptors are ligand-gated ion channels that mediate direct transmitter action. They're also called ionotropic receptors, and they're made up of several protein subunits that are arranged in a rosette around a central pore. So as the ligand binds to one of the receptor subunits, the protein's shape will change. This event opens up the central channel and allows for ions to pass inside the cell. And this is what results in the membrane potential of the target cell to change. The channel-linked receptors are always located opposite of where the neurotransmitters are released from. Their ion channels open instantly upon ligand binding and remain open a millisecond or less while the ligand is bound. At excitatory receptor sites such as nicotinic acetylcholine channels and receptors for glutamate, aspartate, and ATP, the channel-link receptors are cation channels that allow small cations such as sodium, potassium, and calcium ions to pass. However, sodium influx contributes most to the membrane's depolarization. And channels linked to receptors that respond to GABA and glycine allow for chloride ions to pass through, which mediates fast inhibition or hyperpolarization. So this illustration goes over what we just learned about these channel link receptors. So here we have your protein over here, and on these proteins, these are the receptor sites for the ligands. And as you can see right now, this protein is closed. This channel is closed. So these ions that you see up over here that are represented in this yellow, they're not able to move inside the cell. So this is the outside of the cell, this is the inside of the cell. So these guys, they want to come inside. They can't come inside because this channel is closed. Now when you see over here in this image, these green things are the ligands. So for example, maybe this is acetylcholine. So when this acetylcholine comes and binds to this receptor site, now keep in mind that this receptor is very specific to only this specific ligand, okay? So this receptor here only accepts acetylcholine. Another receptor may be sensitive only to GABA, for example. Okay, so that's something else to remember. So in this case, since we're talking about acetylcholine, when acetylcholine or acetylcholine comes and binds to the site, it's going to cause a change in the shape of this protein. And what this does in return is that it opens up these ion channels. Now you can see that these ions are moving inside of the cell. And this is what's going to cause the change in the membrane's potential. G protein link receptors are almost opposite of the channel link receptors we learned about. In G protein link receptors, the responses are indirect, complex, and slow. However, their effects are often prolonged. The receptors are transmembrane protein complexes. They include muscarinic acetylcholine receptors and those that bind to biogenic amines and neuropeptides. Because their effects tend to bring about widespread metabolic changes, G protein link receptors are commonly called metabotropic receptors. When a neurotransmitter binds to a G protein link receptor, the G protein is activated. Activated G proteins typically work by controlling the production of second messengers such as cyclic AMP, cyclic GMP, diacylglycerol, or calcium ions. These second messengers in turn act as go-betweens to open or close ion channels or activate kinase enzymes that initiate a cascade of enzymatic reactions in the target cells. Some second messengers activate or inactivate other proteins, including channel proteins, by attaching phosphate groups to them. Other second messengers interact with the nuclear proteins that activate genes and induce synthesis of new proteins in the target cell. Alright, so in this image here, we're going to be looking at the G protein linked receptors or the G protein coupled receptors. So what you can see is that we have a series of steps that occur over here. And it may look complicated, but it's not that bad. Don't be afraid to rewind it and look at it a second or a third time. But let's break this down and then you're going to understand what's happening over here. So this is a receptor over here. This is the G protein over here. Okay. Now this receptor, it's specific to a neurotransmitter, and this is the binding site for this receptor over here. So when the neurotransmitter comes and binds to this binding site, it's going to activate this receptor. When this receptor is activated, then what it's going to do is that it's going to activate the G protein. Now the G protein, it uses this GTP. GTP is somewhat similar to ATP in that it's a fuel, it's an energy source. GTP stands for guanosine 5-triphosphate. So this GTP is going to fuel, it's going to push this G protein, it's going to give it the energy that it needs, and it's going to cause it to activate adenylate cyclase. So this is an enzyme, okay, as you can see when you look at the name, ASE. So ASE, anything that ends in ASE is an enzyme. So the adenylate cyclase is going to take ATP and convert it into cyclic AMP. Now cyclic AMP is what's called the second messenger, and this cyclic AMP will do one of three things. The first thing that it could do is that it may change the membrane's permeability by opening or closing these ion channels. So remember, when the ion channel is closed, whatever is outside of the cell cannot enter the cell, and things that are inside the cell, ions that are inside, cannot escape. So remember, what do we have? Usually potassium here. We have a lot of sodium and chloride ions that are outside. So when they're open, again, sodium is able to come in, potassium is able to go out, etc., etc. Now, when we go to the second step, part B, all right, the other thing that could happen that cyclic AMP can do is that it could activate enzymes. And usually these are protein kinase enzymes. Protein kinase enzymes will add a phosphate to the protein, which activates the protein. And the third thing that cyclic AMP will do is that it'll go to the nucleus where it activates specific genes, which end up making new proteins. So that's it for these steps. So again, it looks more complicated than it actually is. So if you're going to run through it one more time, remember, we end up getting a neurotransmitter that attaches to this receptor. The receptor gets activated. the receptor then activates the G protein. The G protein then goes over to the adenylate cyclase, and the adenylate cyclase will then convert ATP to cyclic AMP. And then the cyclic AMP will do one of three things. It'll either activate enzymes, or activate a gene, or it'll change the membrane permeability by either opening or closing ion channels. When we're talking about neural integration, we're referring to these groups of neurons and their patterns of communicating with other parts of the nervous system. And these groups contribute to broader neural functions. And remember, we have over 100 billion neurons in our brain alone. And they all have to integrate so that the individual parts, they fuse to make a smoothly operating whole. All of the neurons in the CNS are organized into neural pools. These are functional groups of neurons that integrate incoming information received from receptors or different neural pools and then forward the process information to other destinations. In a simple neural pool, one incoming presynaptic fiber branches profusely as it enters the pool and then synapses with several different neurons in the pool. So when the incoming fiber is excited, it's going to end up exciting some postsynaptic neurons and facilitate others. Neurons most likely to generate impulses are those closely associated with the incoming fibers, and the reason being is that they receive the bulk of the synaptic context, and these neurons are said to be in the discharge zone of the pool. The facilitated zone is where the neurons are on the periphery of the pool, and they're the farthest away from the incoming fibers, so they're not usually excited to threshold unless they're stimulated by another source. So in this image here, you can see the presynaptic fiber over here, and then this is the discharge zone. So again, these are closest to this presynaptic fiber than these neurons over here that are much farther away from this presynaptic fiber. So they're in what's called the facilitated zone, over here and over here. So they're off to the periphery, off to the sides of this presynaptic fiber. In serial processing, the input travels along one pathway to a specific destination. So one neuron stimulates another one, which stimulates another one, and on and on. It's a system that works in the all-or-none manner to produce a specific anticipated response. And the best example of serial processing is a spinal reflex. Reflexes are rapid automatic responses to a stimuli in which a particular stimulus always causes the same response. Reflexes occur over neural pathways called reflex arcs that have 5 essential components: a receptor, sensory neuron, CNS integration center, motor neuron, and an effector. So here you have a receptor over here. The receptor is connected to the sensory neuron. The sensory neuron goes to the integration center, which in this case is the spinal cord, which then turns into and connects into a motor neuron. The motor neuron then goes to an effector where it brings about a response. In parallel processing, inputs are segregated into many pathways, and information delivered by each pathway is dealt with simultaneously by different parts of the neural circuitry. So one stimulus promotes numerous responses. So for example, you're smelling a strawberry, okay? The strawberry is going to be the input. This could cause you to remember when you were a child and you used to go pick strawberries at a farm with your parents. Or it may remind you that you don't like the taste of strawberries. Or it could remind you that you bought strawberries from the farmer's market a week ago. So again, you can see that all these things, all these thoughts, they come to mind with a single stimulus. Okay, that single strawberry brings about multiple responses. So as you can see, parallel processing is important for higher level mental functioning. Individual neurons in a neural pool both send and receive information, and synaptic contacts may cause either excitation or inhibition. The patterns of synaptic connections in a neural pool are called circuits, and they determine the pool's functional capabilities. We have 4 basic circuit patterns, which are diverging, converging, reverberating, and parallel after discharge. So this is an example of a diverging circuit. So here we have a single input, and this single input branches off into multiple inputs. So the farther along you go, each branch, it ends up getting larger and larger. So you can see we go from 1 to 2, and each one of these 2 now connects with 2 more, and each one of these ends up connecting with 2 more. So it's almost like a pyramid if you kind of look at it. So an example is, again, a single neuron in the brain can activate 100 or more motor neurons in the spinal cord and thousands of skeletal muscle fibers. So these are referred to as amplifying circuits, and these circuits are common in both sensory and motor systems. The pattern of converging circuits is opposite of the diverging circuits. However, they're also common in both sensory and motor pathways. In a converging circuit, the pool receives inputs from several presynaptic neurons, and the circuit has a funneling or a concentric effect The incoming stimuli may converge from many different areas or from one area, resulting in a strong stimulation or inhibition. Convergence from different areas help to explain how different types of sensory stimuli can have the same ultimate effect. So for example, seeing a baby smile, smelling their dirty diaper, just joking about that one, smelling a baby after they take a shower, and also hearing them Google and Gaga, these can all trigger a flood of loving feelings in parents. In a reverberating circuit, the incoming signal travels through a chain of neurons, each of which makes collateral synapses with neurons in a previous part of the pathway. As a result of the positive feedback, the impulses reverberate. In other words, they're sent through the circuit over and over again, giving a continuous output signal until one neuron in the circuit fails to fire. These circuits are involved in the control of rhythmic activities such as the sleep-wake cycle, breathing, and certain motor activities like when your arms are swinging when we walk. In parallel after-discharge circuits, the incoming fibers stimulate several neurons arranged in parallel arrays that eventually stimulate a common output cell. Impulses reach the output cell at different times, creating a burst of impulses called an after-discharge that lasts 15 milliseconds or more after the initial input has ended. This type of circuit has no positive feedback, and once all the neurons have fired, circuit activity ends. Parallel after-discharge circuits may be involved in complex, exacting type of mental processing, such as mathematical calculations. The nervous system originates from a dorsal neural tube and the neural crest formed from surface ectoderm. The neural tube, whose walls begin as a layer of neuroepithelial cell, becomes the CNS. The neuroepithelial cells then begin a three-phase process of differentiation, which occurs largely in the second month of development. First, they proliferate to produce an appropriate number of cells needed for nervous system development. Second, the potential neurons, neuroblasts, become amylotic and migrate externally into their characteristic positions. And third, the neuroblasts sprout axons to connect with their functional targets, and in doing so become neurons. So how does a neuroblast growing axons know where to go, and once it gets there, where to make the proper connections? The growth of an axon towards an appropriate target requires multiple steps and is guided by multiple signals. The growing tip of an axon, called a growth cone, is a prickly fan-like structure that gives the axon the ability to interact with its environment. Extracellular and cell surface adhesion proteins such as laminin, integrin, and nerve cell adhesion molecule, NCAM, provide anchor points for the growth cones, telling them it's okay to grow here. Netrin, epinephrine, and semaphore are chemicals known as neurotropins, which respectively attract, repel, or tell growth cones to stop at a specific point. Throughout this growth and development, neurotropic factors such as nerve growth factor must be present to keep the neuroblast alive. The growth cone gropes along like an amoeba, with oozing processes called flipodia, which detect the guiding signals in the surrounding environment. Receptors for these signals generate various second messengers that cause the flipodia to move by rearranging their actin protein cores. Once the axon has reached its target area, it must select the right site on the target cell to form a synapse. Special cell adhesion molecules couple the presynaptic and postsynaptic membranes together and generate intracellular signals that recruit vesicles containing preformed synaptic components. This results in the rapid formation of a synapse. In the brain and spinal cord, astrocytes seem to provide both physical support and the cholesterol essential for constructing synapses. Of all the neurons that are formed during the embryonic period, perhaps 2/3 die before we're born. If axons do not form a synapse with their targets, they're triggered to undergo apoptosis, which is programmed cell death. Many other cells also undergo apoptosis during development. Neurons are amylotic after birth. However, there are a couple of special neuronal populations that continue to divide. Those are the olfactory neurons and some of the cells of the hippocampus. This is the part of the brain that's involved in learning and memory. One of the things that I'm going to argue about is this point over here, where it says neurons are amitotic after birth. From what I've been taught, and this is from higher level classes, is that neurons are mitotic up until the age of 2. After that, they're amitotic. So the question is, what happens as we're learning more? Doesn't the number of neurons increase? And again, the answer is no. What increases are the number of connections between the neurons. So what I would do is go and speak with your professor and find out what they say for testing purposes. Again, you don't want to put down the wrong answer and then, come back to me and tell me, oh, you said up until the age of 2 and then on the test I got it marked wrong. So speak with your instructors. You can tell them that, I said up until the age of 2. Again, this is what I've been taught. I believe this information is not accurate. So in this picture over here, this is an image of a neuronal growth cone. And these fluorescent stains are showing you the locations of the cannabinoid receptors, which are in green. And you can see them all around over here. Also, we see tubulin, which is in blue. In addition to that, we also see the actin, which is in the pink over here. And that's it for Chapter 11, guys. So if you like this video, please be sure to smash that thumbs up button. Also, be sure to subscribe if you haven't subscribed already. Let your friends know, let your classmates know, let your professors know, let your neighbors know, anybody you think that may find helpful. Um, Again, if you have questions, please shoot me an email out. You can find my email address in the description below. In addition to that, you can also leave some information in the comments. I'll try to answer you in the comments area as well. Now, I can't answer you guys all the time in this comments area. I have no idea why, but a handful of you guys have asked me some questions. I try to reply back, and I wasn't able to do it. So if you haven't heard back from me, that means I can't respond to you for whatever reason. So shoot me an email out. You can find my email in the description below. Also, please be sure to follow me on Facebook if you haven't done so already. When I post new things, you'll get notifications right away through the Facebook. So again, thank you so much for watching. Have a wonderful day.